Hello, my name is Alicia Macan Schönleben. I'm ESR number 9, and my PhD project is going to focus on the identification of contaminants of emerging concern in novel foods for human consumption. And I'm doing this at the Toxicological Center at the University of Antwerp in Belgium, and my supervisors are Professor Kovaci, Professor Van Nuys, and Dr. Pomer. Before I tell you more about my project, I'd like to say a few words about myself. So I'm from Nuremberg, that's in Germany, and I did my bachelor's degree in molecular science at the Friedrich-Alexander University in Erlangen, which is very close to Nuremberg. Afterwards, I did a practical semester at the University Clinic, also in Erlangen, and then I did my master's degree in applied chemistry at the Nuremberg Institute of Technology, and as a part of that, I did my master thesis at the Department of Pharmacology at the University of Gothenburg in Sweden. And as I said, I'm currently doing my PhD at the Toxicological Center at the University of Antwerp here in Belgium. So before I started with my PhD, I also worked on a couple of other projects. The first one was my bachelor thesis at the Department for Bioinorganic Chemistry at the Friedrich-Alexander University, where I synthesized the HNO donor and jelly salt. During my practical semester, I then worked on a detection method for the chemotherapeutic drug 5-fluorouracil and uracil and their metabolites in serum and hair by HPLC, and that was at the central laboratory of the University Clinic in Erlangen. During my master's, I then worked on projects at the Analytical Biochemistry Lab at the Nuremberg Institute of Technology, where I worked on detection methods for nicotine and its metabolite cotinine in serum and hair by HPLC. And during my master thesis, I then investigated the in vitro metabolism of the hallucinogenic drug NN-dimethyltryptamine, where we did in vitro enzyme studies and afterwards analyzed by LC-MS. So what motivated me to apply for this training network? Firstly, I would say the project itself, because I've always been interested in toxicology, and later I then found out that I'm also interested in understanding and assessing environmental risks, and I thought the project would be a perfect combination of these two research interests, and it would also allow me to strengthen my skills in these fields. And on the other hand, the training network itself, because one reason is the structure of the training network. It's consisting of academic and non-academic partners, which allows you to do secondments at these partners and workshops. There's also a focus on transferable skills, and the training network provides a scientific platform. So as I said, my project is going to focus on the identification of contaminants of emerging concern in novel foods for human consumption. So since I don't know if everyone's familiar with the term novel food, I'd like to read a quick definition provided by the European Union. So novel food is food that has not been consumed to a significant degree by humans in the EU before May 15, 1979, when the first regulation on novel food came into force. So basically anything that has not been consumed before 1997 in larger amounts in the European Union. So there are different categories for the novel food. So there's innovative food, food produced by new technologies, for example, food that has been UV treated, and food that has been traditionally consumed outside of the EU, so in other third countries. And examples for these novel foods would be edible insects, seafood products like algae, beehive products, or cultured meat, but also plant-based meat alternatives. And before these foods can then be marketed on a larger scale in the EU, they have to undergo safety assessment. And the other thing mentioned in my project title are the contaminants of emerging concern, or emerging contaminants. So that refers to different chemicals, but generally these are pollutants that have a potential risk for humans and or the environment. For example, pharmaceuticals, personal care products, household cleaning products, industrial chemicals, or agricultural products. And little is known about their occurrence and their actual risks, and they're also not regulated under current environmental laws. So my project is going to include the following work packages. First, I will be developing analytical and bioinformatic workflows for suspect and non-target screening methods based on high-resolution mass spectrometry for the chemicals that are available in the database. So for the non-target screening, I won't have any a priori defined suspects, and for the suspect screening, I will, depending on the food, develop suspect list. Then I will be characterizing these emerging chemicals by high-resolution mass spectrometry in the novel foods, and afterwards I will do a chemometric processing of the data to critically assess the performance of these analytical platforms and generate databases, and to establish, for example, any geographical patterns. And then in the end, the food safety of these newly identified contaminants of emerging concern can be estimated in the novel foods. As part of my PhD program, I will also do different secondments. So the first placement is going to be at target analysis in Thessaloniki in Greece in month 10 to 11. The second one is at food forensic, which is in the UK, month 15 or 16. And the last one is going to be at the Josef Stefan Institute in Slovenia in month 32 or 33. So thank you very much for your attention, and if you have any questions, please feel free to ask.